Today we are going to discuss on the chapter the fundamental unit of life, or you can say the basic unit of life. The basic unit of life, okay? Now, this chapter deals with the cell. The fundamental unit of life means the cell. So, we have considered, so cell is considered as the basic unit. Why? Because cell is the structural and functional unit of life. So, the two terms I have used for defining it, that is the structural and the functional unit of life. So, cell can be defined as the structural and functional unit of life. Now, a scientist, Robert Hooke, he in the year 1665, so long back, 1665, he observed the slices of cork slices, under his self-made microscope. We call it a crude microscope. And that under a self-made microscope, he saw that the cork is made up of this honeycomb-like compartments. Okay? And at that time, he did not coin this structure, named this structure as cell, but he named it as little compartments. Later on, it is known as cell. So, the cell was first discovered by Robert Hooke in the year 1665. After that, and that is a crude microscope. You remember this word I have used, crude, self-made microscopes, not an advanced. But after years, we have discovered, or scientists have discovered, now we are getting microscope of these microscopes we are using in our laboratory, compound microscope, and in the compound microscope also you can see the cell. Now, a compound microscope you will also handle in your activity, you will do the onion peel. And in the onion peel, after doing and staining the onion peel, if you put it under a microscope, you have seen a microscope, and there is a stage in the microscope, and you have to put your eye on that lens to see that cell or that slide what you have taken. And you will see that in an onion peel, the cells are arranged like this. There is at the center, there is a nucleus, or at the side there is a nucleus, the periphery there is a nucleus, and there is a place surrounding it. So after Robert Hooke, it was Leeuwenhoek who has seen a cell in a developed microscope. There are many scientists who have discovered, Robert Brown, who discovered the nucleus. So many scientists have discovered cell organelles, and they have coined the name of that cell organelle. But after Robert Hooke, Leeuwenhoek, three scientists, Schleiden, Schwann and Richard, they have proposed the cell theory. What does this cell theory state? The cell theory states that an organism is made up of cells. Cells are the structural and functional units of life, and each cell develops from a pre-existing cell. The word is pre-existing cell. And new cell develops from a pre-existing cell by the process of cell division. So cell theory, 3 points in the cell theory proposed by Sean, Shridan and Richa, the 3 points I told you. Now, what are the 3 points? Now, cell is a microscopic structure. Are all organisms made up of more than one cells? No. There are many organisms who are made up of only one cell. And that cell, according to the definition, that cell forms the structure of the organism as well as that cell performs all the vital functions of the. Those organisms like amoeba, bacteria, paramecium, they are known as unicellular organisms. A single cell forming the structure and performing all the vital functions of the body. But organisms are also there who are composed of more than one cells. We call them multicellular organisms. So we are also multicellular. A dog, a cat, an earthworm, a leech, they all are multicellular organisms, many cells forming the structure as well as they are controlling or performing all the vital functions of the. Now, shape. What is the shape of the cell? All organisms have cell. All the cells are of the same shape and size. No. The shape and size of the cells depend on the specific function. For example, I am giving you 3 examples. See, I am taking 3 cells, RBC, WBC and neuron, the nerve cell. RBC is a blood cell, WBC is also a blood cell and neuron is a nerve cell. Okay. What is the structure of an RBC and how it is related, that structure is related to the function of RBC. Now the function of RBC, we all know that the function of RBC is to carry oxygen. And oxygen they are carrying throughout our body and giving that oxygen to our body cells so that our cell can perform the function. That function is called respiration to obtain energy. So this RBC is very important for us because they are carrying the oxygen, giving to the cell for getting energy. Now, what is this shape of this RBC? RBC is biconcave in shape. It's biconcave in shape. And you all know that you have an idea. A cell has a nucleus at the center. This RBC is actually without, when an RBC gets matured, the RBCs, their nucleus get degenerated. Their cell organelles, they don't have any cell organelles like mitochondria, Golgi bodies, etc. And they are non-nucleated with maturity. Why? Because this loss of nucleus, you can see, if I rub this one without a nucleus, this loss of nucleus increases the surface area, So more is the surface area. If there is more surface area, the RBCs can take more oxygen. They can take more oxygen. And so that more oxygen will reach to our body cells to perform the function that is called respiration. That is called respiration to get more energy. So RBCs must carry more oxygen. So if there is a nucleus at the center, the area becomes less for accommodating oxygen. Therefore, they are denucleated to get more surface area for absorption of more oxygen. This is one example. Next example, let us take the next blood cell example, that is WBC. The WBC, if you see the structure of WBC, it is also a blood cell. If you see the structure, you will see that WBCs, they don't have a specific body shape. They are irregular in body shape, and you can also use the term they are amoeboid in shape because amoeba, they don't have any specific body shape. The same way, WBC, they don't have a specific body shape. Why? What is the advantage? Because as they are irregular in body shape, they can squeeze out through the blood capillaries. This is a blood capillary, and suppose here is an WBC. They can squeeze out through the blood capillaries. They can squeeze out through here by changing its body shape like this. Why? Why do they want to squeeze out? Because they want to engulf germs. To engulf the germs. Here is a germ I have drawn, suppose, in our body, a germ. And this germ is to be eaten up. We are using the term engulf. So this WBC are changing the shape, reaching here and engulfing the germ. If they have a specific body shape, they cannot move out through the blood capillary. So the WBCs are amoeboid or irregular in shape, so that they can reach any remote places of our body to engulf the germs in order to protect us. So you can see the function of WBC is to give us protection. How it is related to the structure of the WBC. Next one, the neuron. The neuron or the nerve cell. How is the structure? You can see the neuron is an elongated, it has many branches. Some branches are short, some branches, one of the branch is long. Why? Why the shape is elongated? Because the function of this neuron is to carry message or to carry the impulse. So to carry anything, if it is elongated, it is advantageous. So these three examples, one more I am adding here, a sperm. A sperm is a male gamete. A sperm the sperm moves. Okay, a sperm moves, and since it can swim, it has to swim, so it has a tail, and that tail helps it to move. So we can say that it is a motile cell. Motile means the one which can move. So RBC, WBC, neuron, sperm, I have given you these four examples, and how their structure is, their shape, their size is related to their specific function. So if it is asked, justify the statement, you can answer this question by giving these examples. Now, children, next I will be coming to the structure of a cell. The structure of a cell, what's the basic structure of a cell? Any cell is having two basic things structurally. One is the outer covering of the cell, and the other one is the inner content, what is composed of. It's composed of inside. So outer covering of the cell, outer covering of the cell and the inner content. Here is the inner content. Okay? And these are the outer covering. Now, this inner content, the whole of the inner content is known as protoplasm. Called protoplasm. And what is the outer covering? First I will tell what is the outer covering. It is present, the outer covering, it is present in all cells. That is the plasma membrane or the cell membrane. Plasma membrane or the cell membrane. So outer covering which is present in all cells, a bacterial cell, a plant cell, an animal cell, all cells. That is the common one, that is the plasma membrane or the cell membrane. But there is an additional outer covering in case of bacteria and in case of plant cell. So in case of bacteria And in case of plant cell, you will find there is an extra outer covering surrounding the plasma membrane. That outer covering is called the cell wall. Now this composition of cell wall is different. In plant cell and in bacterial cell, you will find the composition is different. Now this plasma membrane, which is the outer covering of the cell, of any cell, I will now discuss about the plasma membrane. Plasma membrane. First point, number 1. I'll be giving you point wise because all the time you will find there's a common question, what is the difference between, what are the differences between plasma membrane and a cell wall. So let us do it point wise. Number 1, plasma membrane, is it dead or living? It is living. Is it present in all cells? Yes, present in all cells. Plant, animal. Is it composed of what? What is the composition of this plasma membrane? The plasma membrane is composed of proteins and lipids. Proteins and lipids. Number 4. You will get the term that a plasma membrane is a semi-permeable membrane. So the 4th point, it gives shape to the cell, it gives, you can write also it gives protection. Now, semi-permeable membrane, what does this word mean? Now, inside the cell, substances move in, substances move out. So this plasma membrane allows some substances, not all, to move through it inside the cell or from inside the cell outside the cell. That's why it is not allowing all substances, but it is allowing some substances. So it is called a semi-permeable membrane. Now, this concept is needed for the next topic. So plasma membrane is a semi-permeable membrane because it allows some substances to move inside the cell, also from inside to outside the cell through it. Now, what are the different ways and what are the substances that are moving in and out? Water is moving inside the cell as well as outside the cell. Carbon dioxide, oxygen moving inside the cell, outside the cell, also food. So there are different mechanisms or ways for this transportation. So what are in some transportation, you will find that they need energy to perform this function of transportation, and in some cases you will find very less energy is needed. Now, what are these methods or ways of transportation? Number 1 is osmosis, which you are going to study. Number 2 is diffusion and movement of food in amoeba, endocytosis. So, ways of transportation through the cell membrane, osmosis, diffusion, endocytosis. Now, coming to osmosis. I will define it. You will get 2 terms. low concentration of a solution. First, I will discuss this, and high concentration of a solution. How can you make a solution? You can make a solution by adding solute in the solvent. So, concentration means how much solute is present in the solution. Is the solvent more or solvent less? Or is the solute more or solute less? So, if it is a lowly concentration or lowly concentrated solution, low concentration of a solution, that means here solvent means the water molecules. Solvent plus solute is a solution. Is the solvent more or the solvent less? So, low concentration of a solution means solvent is more. That means water is more. And solute is comparatively it is less. What does this mean? High concentration of a solution means solvent is less and solute is more. So, it depends on the solvent and the solute. Low concentration from a lowly concentrated solution to a high concentrated. So, these two terms we need right now to explain osmosis. Now, definition of osmosis. Let me first define it. Osmosis is the movement of solvent molecules. It is a process of the movement of solvent molecules. From where to where? From a low concentrated solution to a high concentrated solution. So you have an idea what does the low concentration means? Low concentration means where the solvent is more. High means where the solvent is comparatively less. So it is the movement of solvent molecules from a low concentrated solution to a high concentrated solution through a semi-permeable. So there must be a membrane, and this membrane is a semi-permeable membrane. You can get many semi-permeable membrane when we are doing these experiments. Cell membrane is a natural semi-permeable membrane, but there are others also. Egg membrane, cellophane paper, you are using cellophane paper, it is also a semi-permeable membrane. So if this, suppose this is a membrane and this side of the membrane, this is a semi-permeable membrane I have taken, semi-permeable. This side of the membrane is having low concentrated solution, and while this solution is highly concentrated solution. Now what will happen by osmosis? The water molecules, which is more here, water molecules are more and water molecules are less. Water molecules will move from here to here, and it will be a continuous movement from here to here. Don't think that only the movement is in this. The maximum is from here to here, and the water molecules will continue movement until and unless the concentration of both these solutions is equal, same. So, based on this concentration, we can also use the term tonicity of a solution. So, we will be getting three types of solution. One is hypotonic solution, two is hypertonic solution, and three is isotonic solution. Hypo, hyper and iso. Now, I have taken 1 beaker, 2 and 3. Beaker number 1, beaker number 2, beaker number 3. Okay, and I have put a cell here. Let's take an animal cell. A cell here with a plasma membrane. Animal cell, it has a plasma membrane. No cell wall is present in an animal cell. And here is also a cell, an animal cell. And here is also a cell. A cell we have taken with a semi-permeable membrane in all three. This solution is hypotonic. This solution is hypertonic. And this solution is isotonic. What this word hypotonic means? What is a hypotonic solution? Hypotonic solution means where the solvent is more. Hypertonic means where the solvent is less. I am coming to it afterwards. Now, this cell is also having a solution inside it. It has a solution. But since this is a hypotonic, the outside, it is placed in this solution and this solution is hypotonic. Since the outside solution is hypotonic, that means the solvent is more compared to this, which have a less solvent. What will happen? The solvent molecules will move from this solution to this cell body. Why? According to the definition, the move of osmosis, the movement of solvent molecules from a low concentrated solution to a high concentrated solution. So, hypotonic means where the solvent is more. So, when the solvent is more, its concentration is less. Concentration is less. And this is a hypertonic solution where solvent is less compared to the inside of the cell where the solvent is more. Solvent concentration here is more. So, according to osmosis, the water will move from this solvent concentration where the solvent is more to a solvent where the solvent is less. That means to a hypertonic solution. So, outside is hypertonic solution. So, what happens to this cell? The cell losing water molecules and it shrinks. And what happens to this cell? It is gaining water molecules because it is gaining more water from this solution. Because here the water molecules are more. So, it is gaining more water molecules. It gets swelled up. And isotonic means the concentration of this solution and the concentration of this cell inside is same. Both of their concentration is same. Therefore, there will be no movement of water molecules because both sides, in both, the concentration of water molecules is same. So, there will be no movement. Again, I am explaining this one. Hypotonic solution means where solvent is. Hypertonic solution means where the solvent is less, solute is more. And here hypotonic means the solvent is more, solute is less. Solute is less. So if a cell is put in a hypotonic solution, the cell will gain water. Because outside the cell, since it is hypotonic, its water concentration, solvent concentration here is more, and the water has a tendency to move from its higher concentration to its lower concentration. It means water. So it moves from, the water moves from its higher concentration to its lower concentration. Where is the lower concentration of water? Inside this cell. So water has a tendency to move from hypotonic solution to a hypertonic solution where it is less, and the cell will be gaining water and it gets swelled up. When the cell is having more solvent and it is placed in a solution which is hypertonic. Hypertonic means the solvent is less outside and comparatively the cell is having more solvent molecules or more water molecules. So the cell will, this water molecules from this cell will come out. So that means the cell will lose water molecules when it is present in a hypertonic solution and gradually it shrinks. And when a cell concentration, inside concentration of the cell and its outside solution having equal concentration of solute and solvent, then that means it is an isotonic solution, then there will be no movement of molecules in between the cell and the outer solution. Okay, so hypotonic, hypertonic, isotonic. What will happen to a cell if it is placed in a hypotonic, in a hypertonic solution or in an isotonic solution? Now, one thing, children, if a cell is placed, suppose an animal cell, if it is placed here in a hypotonic solution, this cell will gain water and gain water. Ultimately, what will happen? The cell will burst because it has a capacity. It's gaining water, so it will burst. Keep in mind that I have used an animal cell here. But instead of animal cell, if you are using a plant cell here, the plant cell will also gain water. Water it will gain, but it will not burst. Why? Because it has a cell wall that will prevent it from bursting. So hypotonic, hypertonic and isotonic. So again, osmosis definition. I always say that it is two ways I can define it. One is through tonicity and one is through the movement of only solvent molecules. So it is the movement of water molecules from a hypotonic solution to a hypertonic solution through a semi-permeable. Or it is a movement of water molecules from their higher concentration. There means water higher concentration to their lower concentration. That means lower concentration of water where the water is less in which solution. That's always hypertonic through a semi-permeable membrane. It's better you always use through tonicity. Movement of water molecules or solvent molecules from a hypotonic solution to a hypertonic solution through a semi-permeable membrane. It is the definition of osmosis. We have the function of plant, that the plant root hair cell, they absorb water from the soil. How they are absorbing? The method is this one, only the osmosis. They absorb water when the water is entering inside by the process of osmosis. That is called end osmosis. And when anyone is losing water by the process of osmosis, that is called exosmosis. Endosmosis and exosmosis. Endosmosis is the process by which water molecules enter from the soil to the root hair cells. But root hair cells absorb that water. Since it is a plant cell, does not burst. So the water enters to the root hair cells or to the root from the soil by the process of osmosis. There can be various questions if it is asked that a cell is placed in a 20% sugar solution and like that. You will always consider the when percentage is given as a hypertonic solution. So osmosis. Done. We explained, we know about the osmosis. Now the diffusion. Diffusion means the movement of molecules, especially the gaseous molecules, from their high concentration to their lower concentration. No semi-permeable membrane is but in osmosis, a semi-permeable membrane is required. So basic differences here: the movement of molecules, mainly gaseous molecules, from their higher concentration to their lower concentration. No semi-permeable membrane is needed. An example I am giving you: suppose you have sprayed perfume in a room. After some time, after a few seconds, you will find that if you are outside the room, suppose someone has sprayed the perfume inside the room and you are outside, you will get the smell. After some time, after some seconds of 1 minute, you will get the smell. Why? Because that the molecules of the perfume is moving from their higher concentration. Higher concentration means where you have sprayed it, where the person has sprayed inside the room, and you were outside there was no spray. So it moves from its higher concentration, that perfume molecules, to the lower concentration. Where is the lower concentration? Outside. You are standing outside and you are getting the smell. But there are no membrane in between. You are getting how these molecules, perfume molecules, are moving out. That is also by the method. That method is known as diffusion. In our body, in living body, this diffusion process is very much important for the exchange of gases in the alveoli, means in the lungs. We are taking in oxygen, giving out carbon dioxide, and the blood is taking in oxygen, giving out carbon dioxide, and that is taking place through the process of diffusion. Even in a unicellular animal like amoeba, this exchange of gases takes place by the process of diffusion. I am drawing an amoeba here. This unicellular organism is respiring with the help of oxygen. This one cell is performing the function, and by respiration it is producing carbon dioxide, CO2. And gradually what happens? The concentration of CO2 inside this cell gets more compared to the outside concentration, and here it becomes less. And here the concentration of CO2 becomes more. So what will happen? The CO2 will be moving out of the cell from its higher concentration to its lower concentration outside by the process of diffusion. Okay? So diffusion, we are also performing diffusion for the purification of blood in the alveoli, and that is done by the process of this diffusion, this exchange of gases. And one more method, so osmosis, diffusion, and one more method what amoeba perform to engulf food, take in food. That process is called endocytosis, means taking in of solid material by the process of endocytosis, that is done by amoeba. A food here, amoeba can engulf the food with the help of their pseudopodia by the process of endocytosis. Okay, children, then we have discussed here the cell, discoverer of this cell theory, who discovered the cell first. The shape and size of the cell depends on its specific function and the structure of the cell. Mainly, we have discussed the plasma membrane and the transportation of materials through plasma membrane by three methods: osmosis, diffusion, and endocytosis. So, hypotonic, isotonic, explained to you. So, I think it's all these processes, mainly the osmosis, the semi-permeable membrane, the tonicity, explained. Thank you, children, and have a nice day.